go exclusively in the 5' to 3' direction. We start with a piece of RNA primer, which is colored green here. So let me just make that clear. So this little piece is RNA, and all of the green stuff is RNA, and all of the red stuff is the new daughter strands of DNA, and the black is the original parent strand of the DNA. So we have on this particular strand, we first make the primer RNA, and that gets us down to this step 5. Then we bring in DNA polymerase 3, and that synthesizes DNA continuously along here. This then is the leading strand. We would go along with the helicase and the single strand binding proteins, pulling this apart as we go, but this can just continually go in that direction. However, starting here again at this origin, we don't have an enzyme to go this way, so all we can do is go this way in little pieces. These, as I mentioned last period, are called the Okazaki fragments. So we have to make a little RNA and some DNA, another little piece of RNA, some more DNA. So we're going discontinuously along this so-called lagging strand. Meanwhile, if we start here, this strand is the leading strand. You can see here. So this will just keep going. We keep unfolding the DNA ahead of this, unfiling it, and that will just go continuously. But in this strand, we first synthesize that little piece, then that little piece, then that little piece. Okay? So we always have this leading strand with continuous DNA synthesis and the lagging strand with the discontinuous DNA. Step 6. Then the other enzyme, DNA polymerase 1, removes these RNA primers.
And it comes along here, you can imagine here, this is how that photo needs, and I'll indicate this a little bit later into the slide. It's the main role of the primers, and it does that up here and finally, we still have some little bits. We have an enzyme called a ligase that seals these bits. And all this continues until we get to terminator sites and the E. coli there's 7 pieces with 3 pairs. And then another protein called the protein binds to these terminator sites and ends the whole process. Looking at the circular DNA down here, this is the origin of the replication process, and the termination site is going to be down here. So these are all these termination sites where we end the process. Okay, so that's kind of an overview, and let's begin looking at some of the slides then.
Where are I have a quick question. There are no Okazaki fragments on the top of this, right? They're only on the bottom. Isn't that right?
Well, they're both top and bottom. These are all Okazaki fragments on the lagging strand at the top, and these are all Okazaki fragments on the lagging strand on the bottom. So wherever I have these short little things, those are what we call the Okazaki fragments. Okay? So that's the thing. You're going in both directions. So one strand is leading, the other strand is lagging. But the given position, this part is leading, that part going that way is lagging. Same for the bottom. And that's in consequence of the fact that you're going in both directions.
Okay.
Yeah.
I thought it was synthesized 5' to 3'.
Pardon?
I thought it was synthesized 5' to 3'.
Yeah, any position here, well, this refers to this strand here. It's the strand that's being synthesized that's 5' to 3'. So here we are going 5 to 3. Here we are going 5 to 3, 5 to 3. And again, this was just to fill this in here. This would be 5 to 3 prime. Each of these would be 5 to 3 prime, 5 to 3. Okay, well, let's look now
at some of the details of this. And this is just kind of an overview of what I've already talked about. On the left here, we have DNA of E. coli with the origin up there, the RAC site that I've talked about. And as we proceed, we are synthesizing in both directions. We form these little structures which can actually be visualized by electron microscopy. And so on structure, the blue is the parent, the red is the daughter strand. Here is what's going on in eukaryotic chromosomes that have linear chromosomes that are much larger. There are multiple origins of replication. Each one of these forms one of these bubbles, and again, it is proceed in both directions. The replication proceeds in both directions. Finally, they merge, and we get the two daughter DNA molecules. That's what's going on in the eukaryotic chromosomes. Very similar, but linear rather than circular. Okay, we're going back now to the first step in the replication process. And this is the RAC site, the site right up there. And this has a bunch of tandem array have AT that are AT rich in the AT nucleotides, and has these binding sites here for the DNA protein. And so the DNA protein binds sites, pulls apart two strands of DNA to form this replication bubble, that call a bubble. And this is what it looks like here. This DNA protein, as you can see here, is a hexamer. It is able to bind these 5 sites that have sequences that are recognized by these portions of the protein. And that temporarily unwinds that portion of the DNA. We then have the DNA B protein, which we also call the helicase, and that's the unwinding process even further, as you can see there. This is the double-stranded DNA. Here, the helicase is beginning to pull these guys apart. And once they're pulled apart, we have a single strand binding protein that stabilizes these single strands. And so already we've got 3 different proteins into the DNA A, the DNA B, and the single strand binding protein. Now, this is just to blow up the helicase. We looked at this last period. As you can see, it's made up of 6 subunits, and it uses ATP to thread a single 4 strands of the DNA apart. And so, and it moves along that direction there, helping to force these 2 strands. And this is just what I've already explained. And now here is a list of the 3 DNA polymerases that we find in E. coli. I talked last period in some detail about the polymerase 1. And that was first discovered and studied in detail by Arthur Kornberg, for which he received the Nobel Prize. And the main role of this one is not the main part of DNA synthesis, but rather the removal of these little Okazaki fragments, and especially the RNA primers. I'll talk about the polymerase 2. Polymerase 3 is the major one and the bulk of the synthesis. That's the guy that's going to be moving along this leading strand and the trailing strand, doing the bulk
the DNA synthesis. It has a high processivity. We talked about that last period. That means it clamps onto the DNA and stays there for a long period of time, up to 500,000 nucleotides, whereas the polymerase I has a very low processivity. Now let's look at some of these other activities here. The polymerization 5' to 3', that's the normal DNA polymerase activity that we've talked about. They all have that activity. Let's look at this bottom one now. That is what we call proofreading. And let me just expand a little bit on that from what I said last period. That is a 3' to 5' exonuclease activity. And so if we have a piece of DNA, and I'll use this type of nomenclature to represent this piece of DNA, and let me just put in this much of it here, okay? And this could be a T, an A, and a G. We are synthesizing in this direction here. This is the 5' end. This is the 5' end here, the 3' end here. On the 10th strand, this would be an A, a T, but suppose that this was goofed up and this were a T, because of that keto-enol tautomerism I told you about last period, was in the enol form, and instead of binding an A, it bound T, okay? That happens at a rate of 1 in 10 to the 4th, that type of error. If that happened, that could be lethal to the organism, okay? Suppose we get that mistake in there. Then what happens is that this 3' to 5' exonuclease active comes in, we bring in water, we hydrolyze on, and what we get then is this piece here. The way this just continues off here, that's a big long piece of DNA. So this then is the T here. This would be the A here. We would now have OH here, and this is going to be the phosphate there, the G there, and the OH here, okay? So this is going in that direction, 3' to 5', hydrolyzing that particular phosphodiester bond. We call that the 3' to 5' exonuclease activity, or simply proofreading. And it only does this if this here is incorrect. So let's think about the statistics of this. There is a probability that this T will be in the wrong form, in the enol form, of about 1 in 10 to the 4th, okay? So that would be 1 over 10 to the 4th error rate. That's not good enough for replication. That would cause all sorts of mutations down the line. However, if we have a separate proofreading step, if after making this particular nucleotide, we check it in the microseconds that between the initial synthesis and the checking, it's probably switched back again into the keto form, and there's only a 1 in 10,000 chance of it being in the wrong form for the second time. So the second rate of error is 1 in 10 to the 4th. The overall error rate is then multiplied by the first polymerization step times the proofreading step. That's 1 in 10 to the 8th. And so that is the overall error rate for the replication process, and we get that because of this proofreading. And that proofreading is necessary because of the fundamental chemistry that means that this T base is flopping back and forth all the time between the enol form and the keto form, and there's a 1 in 10,000 chance that it'll be in the wrong form at the moment of replication.
But by having two steps, we get to double check it, and the error rate is then obtained by multiplying the error rates of these two steps together. So this is a very important number. That is an error rate that is low enough that obviously it supports life on the planet. Okay, any questions about that? Well, let's move on then. You'll notice here that all of these enzymes have this 3' to 5' exonuclease proofreading activity. However, only this polymerase 1 has this 5' to 3' exonuclease activity. What that is means it's going to chop stuff out. We'll see that a little bit later here. What it's going to do, it's going to be synthesizing nucleotides in this direction, then chopping out this RNA in this direction here. And that's where that exonuclease activity in the 5' to 3' direction comes in. That is for the purpose of chopping out that RNA primer. It's also used for various repair mechanisms.
Which one was used for that?
The number 1, the polymerase 1. Okay, that has that 5' to 3' exonuclease activity. And so that's what uses that for that chopping out. And we'll see that a little bit later. Okay, we've already looked at that. Let's go back to the polymerase 3, the one that carries out the bulk of this synthesis. That's something that has high processivity. Once it binds, it stays bound, and it just keeps going forever, okay? So it's really good. And the thing that makes it keep going for a long period of time is this little subunit here, which is called the sigma subunit. It forms a little circle that encloses the DNA. So when it binds, it grabs onto that DNA, forms that little circle, and stays glued to that DNA parent strand as it moves along. So that's what that does. Here's another space filling model of that. You can see the DNA down the middle there, and then this dimer of sigma subunits forming this little circle around that DNA, which is the parent DNA, and that just sticks to it like glue and won't let it go. So that's what gives the DNA polymerase 3 the high processivity. Okay, so here now are some pictures of how this thing might operate. That thing that we looked at in the previous slide is called the sliding clamp. That's that little circular thing that sort of grabs onto that DNA and holds onto it very tightly. Here is the rest of the DNA polymerase, as you can see there. And that is what is happening on the leading strand. So that can go continuously. Now somehow what's happening on the lagging strand is going to be different. We have to have the RNA primer, which is colored black in this picture, and then we have the DNA polymerase that's going in that direction there. As you can see, we have the helicase and the single strand binding protein pulling apart the parental strands of DNA as we go along there. Okay, here's a blow-up of what we think this whole complex might look like. And although we have structures of individual parts of this, we don't have a good structure of the whole thing during the replication process, so we're kind of doing some imagination here. But it's thought that we have one subunit of the DNA polymerase 3 sitting here, and it is attached to the other subunit through something that is called the clamp loader, which is colored green here. And then that is attached to this DNA B protein, which is the helicase. So there's this huge complex that somehow it all interacts together to carry out this synthesis on the DNA strand. And here is a representation of one of the current models of this. I emphasize that this is a model. There's certain aspects of this that are not known. You see here the one piece of the one subunit of the DNA synthase enzyme on the leading strand. It's got a straightforward job. It just goes continuously in that direction there. Here we have the DNA polymerase on the lagging strand. It's got a much more difficult job. It has to synthesize these little pieces, then release and start over again. And one proposal is that we form a little loop of the parent DNA and the lagging strand that resembles a slide trombone, okay? And so as the lagging strand comes in here, it puts out a long bubble here, resembling the trombone when you extend it. And then at some point, this DNA synthase gets on that and goes in this direction here. And then it has to release and come back and start over again. And remember that each time it starts over, the RNA polymerase has to get in there as well and carry out that synthesis of that primer. So this is all a complicated process, and this is one picture of how the whole thing might work. So that's what we know so far about it. Okay, now we're getting nearly to the end. We have to remove all of these little primer RNA molecules. And I'm going to go ahead and show you how that is done. So far, all of this has been carried out by the DNA polymerase III enzyme. So now the DNA polymerase I enzyme comes in here, and I better get the right colors here. And what that DNA polymerase does, think of it being right here. It comes in and binds right there, and it moves this direction. It has the synthetase activity, so it can extend this guy here, but it also has this 5' to 3' exonuclease activity, and so it chops ahead of it. So as it's moving along here, what's going to happen is it's going to chop out this RNA polymerase using its 5' to 3' activity, and it's going to move, extending that with DNA. So let me just do a few of those. One of the, and I'll just get rid of that now. It's moving along as this proceeds. Then the same enzyme starts here, moves this direction, chops out that 5' to 3', continues up to there. This part here would do the same thing. We would extend there up to there. Okay? So that DNA polymerase I has the activity to chop up the RNA primers and extend the DNA to fill in the gaps left by those primers. The technical word we give that is nick translation. And this is another picture of the chemistry that's happening up here on the board here. Imagine that's the nick there, and that little piece is RNA, even though they don't show that in this slide here. What the DNA polymerase does is it chops out one of those strands, extends it with newly synthesized strands, it's colored red here, and then the nick comes up there. So in a sense, it translates the nick from there to there, and if there was RNA in that position, it would chop all that out. We still have these little nicks. We still have Okazaki fragments, okay, but we got rid of the RNA at this point. So that's what the DNA polymerase I does. So we're almost done now. The final step here is going to be to use another enzyme called DNA ligase, and that enzyme seals the nick. So that enzyme, the DNA ligase, let me just put that here, DNA ligase, it will come in and seal up all these nicks and make that strand continuous. So that's the function of the DNA ligase. And its structure, or its, now I've lost my pointer, here it is. It is showing up here. It takes the end of a nucleotide that has a 3' OH, that would be one of these little nicks here, and it allows that nick to be sealed that has this phosphate group here on the 5' end, as you can see. It needs ATP in order to do this, and it uses the ATP twice, by first breaking it down to AMP and pyrophosphate, then hydrolyzing the pyrophosphate into 2-phosphates. And it forms the new phosphodiester bond, sealing that nick. So the role of the ligase is to seal the nick between the 3' OH and the 5' phosphate that you see there. So that's all done now. We've sealed all the nicks here. We do the same thing down there, seal all those nicks. We continuously do this until we get to the very last step, and that is to get to the termination step, where we get to the end of the molecule, and then we actually terminate the replication process. Okay, so any questions on that? It's a pretty complicated thing, and there's a bunch of enzymes involved, but they all work together to replicate our DNA molecule. Okay, just one other little thing I'd like to mention. In eukaryotic chromosomes that are linear, we've got a problem because we have these lagging strands, and if we're carrying out replication processes repeatedly, and we do what we've already talked about, there's a leading strand and a lagging strand, but when you get up to this problem here, on this lagging strand, you get a little gap at the very end. You don't have that problem in circular DNA, but in the linear DNA of eukaryotic chromosomes, we have this little gap here. And every step of replication, you have a possibility of that gap getting shorter. And that is another thing that could lead to aging. So there's a little bit of extra DNA that's at each end that is called the telomere, that gives you kind of an overhang here, and that telomere protects the DNA from this progressive loss of the ends due to this leading and lagging strand type of problem. And this molecule, this end here, is kind of rich in G, and it is thought to pull back upon itself and kind of protect the ends of the DNA from being progressively chopped away by this leading strand, lagging strand type of problem. And there's a lot of interest in these telomeres and how they affect aging and how they affect cancer. Cancer cells, as you know, have to replicate very rapidly, and so obviously their telomeres would limit their ability to replicate, and so there's enzymes that kind of go out of control and produce extra telomeres in those cancer cells. Okay, any questions on this then? Okay, well, let's have a short break, and we will begin the transcription study then.